Okay. So what I want to go over today is just some stuff on genetics. Um, so we're going to be doing more of these SNP tests, which stands for single nucleotide polymorphism. It means that there's a glitch in one of the genes. So I had purchased these books. They're pretty good books. This is Clinical Genetics Made Ridiculously Simple. Just takes you through the basics of genes again, just to kind of refresh myself and a few things. So when they tell you about the cell, you guys probably remember learning this in biology. When you learn about the cell, they tell you that there's the cell wall, and so the cell is the smallest living thing. And then within the cell are things called organelles, like the Golgi complex, rough endoplasmic reticulum, the vesicles, the cytoskeleton, the cell membrane, the smooth endoplasmic reticulum, the mitochondria, the ribosomes, and all these components make up the cell. And when the genes were discovered, which is this right here, they basically said that's the beginning of everything. So they thought that the genes ran the show, and that the genes were the basics of the cell working and producing and living, and that if there was glitches in the gene, that there's nothing you can do. And then through the 5 years after they discovered genetics, they come up with a term called epigenetics, which means above the genes, the things that are turning the genes on and off, or that could influence the genes. So from 1990 through 2002, the U.S. government and a few other companies basically said they're going to go into this genome here, and they're going to map out the whole human being. And then they're going to find the glitches, and we're going to be able to fix the glitches because the genes are the beginning, right? So if we can find the bad gene in Jaron for his allergies, we can change the gene and we'll get rid of his allergies. And we can change the gene and get rid of whatever it is. The problem is that they went in and they said, okay, there's about 130,000 proteins that are made inside the human body. So if the gene is the beginning starting place, we should find about 130,000 genes, right? If this gene encodes for that protein, and we know the number of proteins, about 130,000, we should find 130,000 genes. So within 2 years of doing the mapping, the laying out of the human genome, they basically come to the conclusion that there's only about, this is the geneticists, by the way, they come to the conclusion there's only about 20 to 25,000 genes. So the genes cannot be the beginning of anything, because there's only 25,000-ish genes, and yet there's 130,000 proteins. So the genes will turn on and turn off based on epigenetics, what you do right, what you do wrong. So that's most important to understand. And then just some basics. There's 23 chromosomes. So there's a chromosome is like DNA. The total DNA on all the chromosomes is 3 billion of these base pairs. A single gene will have approximately 27,000 base pairs, and hundreds to thousands of genes are on each chromosome. And then the human has about 20 to 25,000 genes. So as part of this chromosome, you then have the genes, and then as part of the gene, you have these base pairs, which are called nucleotides or nucleoside. So when you get a glitch in one of those called single nucleotide polymorphism, that means that it could change the function of that gene to either not work at all, work too much, or be a little slow. But when it's all said and done, what percentage of diseases are truly genetic? 5%. So this is not the starting place whatsoever. But we're going deeper into this now to look, because if I find out with whoever that within this biochemical pathway, Jaron has 2 genes that are slow, we can do certain things to help the slower genes. And again, you're not going to cure major disease, but you can definitely influence the biochemistry and the flow of things over a time frame. And then when you're talking about biochemistry, there's things called substrate. So there's the substrate is this, then there's the enzyme this, and then there's the product, which is this, okay? So the enzyme, these are located all throughout your biochemistry, and we have charts in the back that say, hey, here's this pathway, and throughout this pathway there could be 20 enzymes, and within those enzymes are genes. And if we find a single nucleotide polymorphism, meaning a glitch in that gene, the gene may work not at all, or slower, or possibly faster. And then I can understand the biochemistry and the flow of all of this that's happening within the cell, the whole cell here. So one more thing on this that's important. So we can enucleate the cell, meaning take out the nucleus and the cell stays alive. So inside the human body, if I take out your brain, you're dead instantly. The brain is the master controller of the body. That is the master system, right? The brain. Well, we thought the nucleus was like the brain of the cell, except you can cut out the nucleus and the cell keeps living for a while. The cell will keep living. So it's not the nucleus. So what it is, what the brain of the cell is, this right here, the cell membrane. And the cell membrane is made up of fats, cholesterol, and your essential fatty acids. So if you eat garbage and you're taking low cholesterol foods, or you're taking drugs to lower cholesterol, you will totally negatively impact every single cell in your brain and your body. So, oh yeah, here's one more thing too. So how many chromosomes does the human have? 23. Okay. The largest is number 1. And on the number 1 chromosome, there's about 8,000 genes. The smallest is number 25, and there's 225 genes on that. And remember, there's a single gene will have 27,000 base pairs. So if you have 8,000 genes times 27,000, that's how many base pairs or SNPs you could have on the first chromosome. So when we do these testings now on these SNPs, we're measuring various different enzymes, and then the genes in the enzymes, and then the nucleotides in the genes to say, hey, is there a SNP? And based on the Human Genome Project, we know the biochemistry. We go, okay, so that's the enzyme that drives that process. And in that enzyme, there could be a SNP in that enzyme that's not letting it work optimally. And then we can get the genetic testing and look at that. It's very complex, and get the genetic testing and look at that to say, how could we support that better? The other big thing that's important is your genes down in the chromosome and then down into your DNA. Your genes are covered by a thing called your methyl group, CH3. So I always picture this, that your genes are up on the wall like letters, A through Z. And if the gene is covered, it cannot express itself, meaning it doesn't do anything. It's covered. It's turned off. If I uncover it, the gene expresses itself. Start there first. Go after the physical, chemical, mental, emotional stressors. Clean up what the person is doing. Look at how the nervous system is working. Look at their cellular energetics. Look at the lifestyle, foods, and nutrients. And after that's going, then we can talk about this genetic thing. But you shouldn't jump in there. It's downstream information. You got to start with the other big stuff first. And once that's cleaned up, you can go deep into looking at what's going on there. Okay, so that's the quick little rundown of the genes.